Hello guys, so we'll discuss next question, which is in continuation with principles of heredity and variation. So we discussed about test cross and back cross. Okay, 23rd question, test cross in plants or in Drosophila involves crossing between two genotypes with recessive traits, between two F1 hybrids, between two F1 hybrids. The F1 part, the F1 hybrid with a double recessive genotype. Double recessive means recessive like this. And between the D part is between two genotypes with a dominant trait. So C test cross is related with like F2 word. So that is to determine, or F2 means to determine the genotype. Since it's written here. To determine the genotype of a tall plant at F2. So here we are not seeing any F2 word here. So here test cross involves crossing between two F1 hybrids, and C part is the F1 hybrid with a double recessive. Double recessive means it should be in pure form. If it's written only recessive, that means it could be like this also. I mean, it will be capital T and small t. It could be capital T and small t. So if this is C part. Next is okay. 24th question. ABO blood groups in humans are controlled by gene I. It is 3 alleles I, IB and I. Since there are 3 different alleles, 6 different genotypes are possible. How many phenotypes can occur? Phenotypes. Okay, so see, this is part of codominance blood grouping. IA, then IA will give you A blood group. IA and I will again give you A. IB and this will give you B. I will give you B, again B. IB will give you AB. I will give you O. Since there are 4 phenotypes, 4 blood groups, so here answer is 4. Genotype plant should be dominant. Phenotype can be determined by showing the dominant here. So since here in NCRT there's a term that's called a test cross. So that's written to determine the genotype of a plant at F2. This is the typical line of NCRT, that is test cross. You can see here, to determine the genotype of a plant. This is paragraph of NCRT. Okay, next question. Which one of following cannot be explained on the basis of Mendel's law of dominance? Cannot be. The discrete unit controlling a particular factor is character is called factor. Then discrete unit. Okay, out of one pair of factor is one is dominant, other is recessive, factor occurs in pairs. uh, Occurs in pairs like this. Alleles do not show any blending and both the characters recover as such in F2 generation. So he didn't talk about blending or any mixing. Like if I talk about blending, mixing means if I take example of incomplete dominance like red and pink flowers, so that was an exception to Mendel's rules. Basically he didn't show any blending. Okay, like you can see law of, if you see law of segregation, that means separation. Separation means like they are having their individual identity. If individual identity will be there, no mixing will be there, they will express themselves. Okay, next, ABO blood grouping is controlled by gene I, which has 3 alleles and shows codominance. There are 6 genotypes. How many phenotypes are possible? Phenotypes, again, this is 4: A, B, AB and O. Okay, next is a cross in which an organism showing dominant phenotype cross with recessive parent in order to know the genotype. So this is the NCERT line to know the genotype. That is test cross. Mono hybrid cross is like TT into TT. Dihybrid cross is like capital RY. 2 factors are there. Then like this and like this, we make a Punnett scale like that. Back cross is with when we cross it with one of its parents. Like back cross is like if I cross F1 with capital T or small tt. This is back cross. Test cross, like when you need to test the cross where determine the genotype. Like you don't know what's the genotype, so I need I don't know what it is here. Violet flower and purple flower. So I know the result is this. So I can make out this could be capital W. So this is homozygous condition. This is to determine when we don't know the genotype. Like test like what it should be here, like that. So answer is C. Okay, coming to next question. In Antirrhinum, 2 plants with pink flowers were hybridized. The F1 plant produced red, pink and white proportion of 1 to 1. What could be the genotype of 2 plants used for hybridization? Red flower is determined by white by RRG. What could be the genotype of 2 plants? So they are saying F1 produced red, pink, white. What is the genotype of plants used for hybridization? So this is, they are talking about pink flowers. What could it should be pink flowers. Okay, next is, this is part of incomplete dominance. In pea plants, yellow seeds are dominant to green. Yellow are, so that means this is yellow dominant to green. Here they are talking about seed color. If a heterozygous yellow seeded plant is crossed with green seeded plant, what ratio of yellow and green seeded plants would you expect in F1 generation? So see, basically you see the main phenotypic ratio is 9331. Genotypic is like 1 ratio 2 ratio 1 2 2. Genotypic is like is very much long. So basically you should know the phenotypic ratio, that is 9331. So this, if we cross it, what we'll get, YY like this, and here it will be Y, here it will be Y. So 2 are yellow, and 2 green. So that means if you see the ratio, that will be half 50-50. So this comes out to be 50-50. Okay, so this was the last question. Thank you.